The unit 1 is diversity in the living world. So under diversity in the living world, you have 4 chapters that are there. Remember, photosynthesis is not just plants standing in the sun and absorbing the sun and by the chlorophyll pigments and converting into chemical energy and making use of it. Question paper pattern. This is very, very important because you study the entire book and you don't know the pattern of the question paper. Reading the entire book is a waste. So, hello everyone, a warm welcome to this session on First PUC Biology. I am Dr. Divya, Biology Faculty, Vidhyaashram Pre-University College, Temple of Excellence, Mysore. So in this session, let us look into the introduction for First PUC Biology, wherein we'll study about the syllabus, what is the textbook preferred, what are the chapters under that particular syllabus, and how the question paper pattern looks like, both the theory as well as the practical, and what all experiments you'll study under the practical classes. So moving on to the textbook. So the textbook that is preferred for First PUC Biology is prescribed by NCERT, that is National Council of Educational Research and Training, NCERT. So you can have your old textbook also, or you can go in for the new print or the new edition because some of the portions are deleted. But you can keep the old textbook and have a look into what are the portions that are deleted and just keep studying those portions from your old textbook. So, having an old version or old edition and keeping a new version, both will help. So, this is the textbook for biology that is preferred by NCERT. And all the diagrams, whatever answers you write, should be from this textbook itself. And also the diagrams, whatever is there in the NCERT, only that should be drawn for the exam. Only those markings should be done. You're not supposed to draw diagrams from somewhere else, though it may be correct. Next, talking about the syllabus prescribed by the PU board. That is there in the NCERT textbook. So, it is divided into 5 units. The unit 1 is diversity in the living world. So, under diversity in the living world, you have 4 chapters that are there. So, first is the living world, wherein you'll study what are the organisms in the living world in general. You'll study about how to name an organism by binomial nomenclature, what are the rules, all that you'll study under the living world. Once you know the rules and how the scientific name is written, then you'll study about biological classification, which is the second chapter. So, under biological classification, you'll study how the organisms are grouped into various classes. So, all that you'll study here. So, for easy understanding, the organisms are grouped into different kingdoms. So, all that you'll study under biological classification. Next, you'll study plant kingdom and animal kingdom separately under chapter 3 and chapter 4 respectively. So, chapter 3, why they have kept plant kingdom separately is because it is very, very vast. Lots of things are there to learn. So, that is why they have not clubbed it under biological classification. It's been kept separately. So, here you completely study about the various classes that come under the plant and examples, all that you'll study under plant kingdom. And in animal kingdom also, you'll study the various classes that come under the animal kingdom or the kingdom Animalia. Next, moving on to unit 2. Which is structural organization in plants and animals. Now, since you know about what are the different plants that are there, how they are grouped, classified, what are the different animals that are there, how they are grouped and classified, then you'll move on to studying its structure. So, all of these chapters are connected to one another. So, first we'll move on to studying the structure of the flowering plants, wherein you'll study morphology of flowering plants, that is chapter 5. So, what is the structure of the flowering plant? What is there in that? The petals, the stamens, the stigma, all that reproductive parts, everything we'll study under morphology of flowering plants. Next, we'll move on to anatomy of flowering plants. So, morphology means when you look at a plant, what all the characteristic features you can see in them, that you'll study under chapter 5. When you move to chapter 6, you'll study deeper, wherein you'll take a section of the plant. When you take a section of the plant, how the cells are arranged. You have parenchyma, collenchyma, chlorenchyma, sclerenchyma, food conducting tissue, water conducting tissue. All that you will study under anatomy of flowering plants. So, inside the plant, how the cells are, what the cells are made of, how the cells are arranged, all that you'll study under chapter 6. Next is, you'll move on to animals, wherein you'll study structural organization in animals. That is the different structures of animals, their muscles, how they look, and all that you'll study under this. Next is, because you know the structure now, you need to know their functions. Therefore, moving on to unit 3, that is structure and functions. So here you'll study about the cell. So both plant cell and animals, they have cells. They're nothing but made up of cells, right? So you'll study that in chapter 8, which is cell, the unit of life. So now you know about cell. So for functioning of the cell, for various pathways to occur in a body, you need to know what are the different biomolecules that are involved there, like fats, lipids, and proteins, carbohydrates. About all that you'll study under chapter 9, that is biomolecules. Now, because you know about cell and you know about biomolecules, they have kept the chapter 10, that is cell cycle and cell division. So in cell cycle and cell division, you'll study about how the cells divide. There are two methods of division. One is called as mitosis and the second one is meiosis. Under mitosis, you have various sets of division, and under meiosis also there are different sets of division. All that you will study. So cells, if they have to multiply, they need to divide, right? How exactly they divide? You will study in cell cycle and cell division. That is chapter 10 under unit 3. Moving on further, now because you know the structure and the function, you need to know the physiology. Physiology means how plants respire, how they photosynthesize, how they transport the nutrients, minerals, all that you will study in this particular chapter. Like some of the topics are deleted under this particular unit, plant physiology. So 3 chapters are remaining under this. So you'll study photosynthesis in higher plants. Photosynthesis is nothing but an autotrophic mechanism, right? So here you'll not just study, remember photosynthesis is not just plants standing in the sun and absorbing the sun and by the chlorophyll pigments and converting into chemical energy and making use of it. Here you study all the chemicals that are involved, or biochemicals or pathways that are involved under photosynthesis. Next you'll move on to chapter 12, that is respiration. Here also various cycles or pathways are there which you need to study under respiration in plants. It is not just respiration in plants means the plants take in carbon dioxide and give out oxygen. No, that is not the case. When during that process of taking in of carbon dioxide and giving out of oxygen, various pathways come in between. So that you will be studying under respiration in plants. Next, in plant growth and development, you will study various hormones that help in the plant growth and development, and also when there is availability of sunlight, how the plants grow, when there is non-availability, how the plants grow, how they cope themselves to grow under dim conditions. So all that you will learn under plant growth and development. So that was about plant physiology. After plant physiology, you will completely study about human physiology. So in human physiology, you'll study about how the breathing and exchange of gases occurs. So here also breathing means it's not just you taking in oxygen and giving out carbon dioxide. There is a process which is connected to the lungs and to the heart during the breathing process, right? So the entire thing, the structure of the lungs, the heart, all that you will study here. Next, you'll move on to studying about, in chapter 15, body fluids and circulation, wherein completely you'll study about heart. The structure of the heart, what exactly the heart does, how it takes in the oxygenated blood, and the deoxygenated blood, how it gives the deoxygenated blood back to the lungs and it is pushed out of the lungs. All that comes under body fluids and circulation. Next is excretory products and their elimination, wherein you'll study about the kidney here in this particular, and how urine is formed, all that. That is nephrons, about the basic units of kidney, that is nephrons. All that you'll study under chapter 16, that is excretory products and their elimination. Next, chapter 17 is about locomotion and movement. So we all are locomotive, that is we are motile, we are capable of moving from one place to another, right? So if we need to move, for example, I'm moving from one place to another here, like how am I able to do it? There has to be some sensory mechanisms that is sending signal to my brain saying that, okay, now I'm supposed to move, right? So how that actually occurs, that you'll study under locomotion and movement. So that is under chapter 17. Next, chapter 18 is neural control and coordination. Now you know that everything in a body is controlled by the nerves, right? wherein there is a coordination between the nerves of different parts of a body with that of the brain. That is with the peripheral nervous system and with that of the central nervous system. So how exactly the messages are exchanged between the peripheral nervous system with that of the central nervous system, that is the brain, which makes us to perform various actions that you will learn under neural control and coordination. So all are nothing but chemicals or synaptic messages that are sent to the brain. So all that we'll study under chapter 18. Next, moving on to chapter 19, that is chemical coordination and integration. In chemical coordination and integration, you'll study about various glands and hormones that are present in the human body, which actually are needed for the normal functioning of an entire body. So all those hormones you'll study under chapter 19. So this is about the syllabus. So totally you have 5 units and a total of 19 chapters under this 5 units. So this is about the syllabus. Now moving on to distribution of marks, like how the marks is distributed in the question paper. What are the exams that you write? So your final exam will be for a maximum marks, that is total marks is 100. In that, again it is split because biology students will have theory as well as practicals. Theory is the one that you write and practical is the one, practical separate practical classes will be held, and in that for that practical classes an exam will be conducted, practical exam, which will carry 30 marks. So theory is for 70 marks, you will be writing for 70 marks and you will be performing the practicals for 30 marks. So therefore total it is 100 marks. And you have to remember some of the general instructions that you need to follow while writing, be it theory or practical. So the question paper, so this is for theory. So the question paper consists of 4 parts, that is part A, part B, part C and part D. Again part D is divided into 2 sections. So look into that later. And all the parts are compulsory. It's not that you can skip any one part because there will not be choices between the parts. Only choices between the questions, or in the one section they will give choices in the mains, not the whole part you cannot skip. So all the parts are compulsory, and wherever diagrams are there you have to draw the diagram, and not just drawing the diagram, based on the marks you have to put label. If it is for 5 marks, at least 10 labeling should be there. If it is for 3 marks, at least 6 labeling should be there. So that is how you have to label. So you draw a beautiful diagram, you spend somewhere 5 minutes drawing a diagram in biology exam and you don't label it, it will fetch you only 0 marks. So labeling the diagram is also very, very important. So you have to draw diagrams wherever necessary. Sometimes they will ask directly, draw a neat label diagram of the heart. They can ask directly for 5 marks. Or sometimes they will not ask, they will ask you to write, explain about heart. So if it carries 5 marks, nothing will go if you draw a diagram. It can fetch you marks, right? So based on, you have to think by yourself, like where the diagrams are necessary, where it is not necessary, based on the marks, even though they don't ask you directly to draw a diagram. So wherever diagrams are necessary, draw it, and labeling the diagram is very, very important, because unlabeled diagrams or illustrations or any schematic representations are there. If you don't label them properly, it will not fetch you any marks. So that is why you have to label the diagram. And also make sure that the diagrams are not microscopic. It is at least a bigger diagram which you draw, at least half a page of your answer script. Please draw the diagram. It will look big and beautiful. So that is how you need to draw. Next is question paper pattern. This is very, very important, because you study the entire book and you don't know the pattern of the question paper. Reading the entire book is a waste. So you have to study based on the question paper pattern. So here, I told you earlier itself, it is divided into that is A, B, C and D, right? Part A, part B and part C, part D. So under part A, 15 questions will be asked. So question number 1 to 15, that is there, it is entirely multiple choice questions, wherein 4 choices will be given. You have to choose the right answer. So this is very difficult, because you have to study the whole textbook for this, because from anywhere the multiple choice questions can be picked up. So understanding the concept is very, very important. Next, question number 16 to 20. You have under part A, there are 2 mains, first main and second main. So earlier the question paper pattern was different. Now it is different. It's changed a bit. So question number 16 to 20 is entirely fill in the blanks. So here you have to choose the appropriate word. Right? So here choosing the appropriate word means already if they ask 5 questions, 5 options will be given. You have to choose the appropriate answer for those 5 questions respectively. So that is how it is question number 16 to 20 which comes under main 2. And make sure that when you're writing, you write the mains and the question numbers. Okay, here if you don't write the main also it is okay because it is in a continuous order, right? 1, 2, 3, 4, like 16, 17, they have continued the questions up till somewhere 30 questions or something like that. So if you write the question number correctly also it is enough, but make sure that you write the question numbers correctly. Otherwise, though you write the answer correctly and you put the question number wrong, the entire answer will be marked wrong. So that is about part A. Next part B, question number 21 to 28. So here some questions will be given. That is 8 questions will be given, out of which you have to answer any 5. So any 5 you have to answer. This is 2-mark question. So part A is entirely 1 mark. Part B is entirely 2 marks. So wherein 8 questions will be given and you have to answer any 5. The number of options is also more for you. Next, moving on to part C. Here question number 29 to 36. Here again it is 3 marks question, wherein again 8 will be given. You have to answer any 5. It is 3 marks question. Next, part D, question number 37 to 42. So here answer any 3. So 6 will be given. So you have to answer any 3 out of it, which will fetch you 5 marks. So it is 5 marks questions will come under part D. So part A is entirely 1 mark. That is section part A, first main and second main. Part B is 2 marks. Part C is 3 marks. And part D entirely it is 5 marks. So earlier it had section A and section B. Now they had made it as 5th main and 6th main. So this is how it is. Question number 43 to 47. Here also 4 questions will be given. Any 2 you have to answer out of it. That will also, it is for 5 marks only. So this is how, and not just that, don't think that if they ask you for 5 marks, it is not necessary like they'll ask only 1 question. They can split it as A and B. So under, say for example, under question number 45, they can split A, they'll write 1 question which can be for 3 marks. Then B, they'll ask 1 question which can be for 2 marks. So total it is 5 marks, right? So like this they can split. Sometimes they can ask 1 mark question only, 5 questions they can ask under part, that is D. So that is why biology you have to study entire book. So you cannot say that okay in 5 marks section only 5 marks will be asked. No, they can split and ask. Even in part C also they can split and ask like 3 marks, right? So 2 and 1 or 1, 1, 1 like that they can split and ask the questions. So this is about the question paper pattern. So moving on to the practical. So you know the syllabus for the theory. Now we'll look into the syllabus for the practical. So now also it has changed from last year. The practicals, like some portions have been deleted and new one has been added. So all those changes are there. What are those? We shall look into. So the first experiment will be to study a compound microscope. So microscope, what are the parts of the microscope? What magnification lens are used? All that you will study under this. And next is to identify and study morphology of bacteria, fungi and different plant groups. So what is the structure of the bacteria, different fungi, that is bacteria, whether they have a flagellae, what is their cell, how it looks, what is the shape, and fungi, talking about fungi, like what is the mycelium, how it is arranged, whether it is cenocytic or all that you will study. So if you know the theory well, practical will also be easy because both are interconnected to one another. And about different plant groups, all that you will study the morphology of the plants also under this. Next, third one, to study some animals on the basis of their external features. This is also again on the morphology only. So studying of animals, so wherein specimens will be kept, you have to study what type of feet they are made up of. Externally, when you look at the animal, what all features you find to see to get some peculiar features, all that you will study here. Next, fourth is study of mitosis. Here mitosis comes under cell division. So under this you have interphase, prophase, metaphase, anaphase, telophase. So all that you will study, different phases you will study under this. Next is to study and identify the inflorescence. So inflorescence is nothing but the Flowering. Flowering means a bunch of flowers. So how the bunch of flowers are arranged in a plant, in a particular plant. What are the different types of inflorescence? Like we have racemose, cymose, spike, spikelet. So all those different inflorescences, names are given for that based on how they are arranged. Umbilical. So you have to study that under this experiment 5. Next is study and describe flowering plants of families. So here Solanaceae family, that is brinjal, potato, about that you have to study. Their floral diagram, that is how the petals are arranged, how the stamens are arranged, how the calyx is arranged, all that you'll study under this there, that is their flowering pattern. How it is arranged, then how the leaf looks like, how the stem looks like, how the entire plant looks like, you'll study under this particular experiment number 6. Next is 7, to study anatomy of stem, that is and root of monocots and dicots. Monocots which have one cotyledon, dicots which have two cotyledon. Grass is a monocot, dicot, most of the flowering plants are dicot. So those plants that are mentioned in the practical manual, that you have to get it to the laboratory, and we will be studying about. We'll take a thin transverse section or a cross section of the stem and the root and observe it under the microscope and study how the cells are arranged. So that is experiment 7. Next is we have to study external morphology of animals through models. So they have mentioned here frog. About only frog we have to study the external morphology. How is the skin? How is the feet? How is its nostrils? So all that, or they don't have ear, tympanum they'll have. So all that you have to study its external morphology under experiment 8. Next is to study the distribution of stomata. So stomata is nothing but that which also helps the plant to breathe or exchange of gases. So there are some kidney shaped structures that are there which you can beautifully look into it under the microscope. So both the dorsal surface as well as the ventral surface of the leaf we have to take and we have to peel it and look for the stomata under the microscope. Next is to detect the presence of carbohydrates like glucose, sucrose and starch. So we'll take a particular green gram or something like that, grind it and put suitable chemicals for that to see whether the different carbohydrates are present in that or not. So this is a biochemical experiment because you have molecules as a theory lesson, right? So that you will be studying here in practicals as well. Next is to detect proteins. Same way, this is also a biochemical experiment, that is experiment 11. Experiment 12 also is a biochemical experiment wherein what are the different types of fats, different animal tissues, whether fat is present or not, we have to, and also different plants and animal tissues, whether lipids of fat is present there or not, we will study under experiment 12. Next 13, wherein we'll study the separation of chlorophyll pigment. So you'll just grind a leaf material, put the pigment on chromatography paper and then put it in a solvent and then see how the pigments get separated. So plants usually have chlorophyll A, B, C, then xanthophylls, carotenoids, all that. So based on the color they separate, we can see what type of pigment is there. So this is a very beautiful experiment. That is experiment 13. Next is to study the rate of respiration in flower buds or germinating seeds. So germinating seeds or flower buds can be used wherein using respirometer we can see how much respiration the plant produces. So that is one of the experiment. Next is various setups will be kept which will be prepared like photoperiodism, about like how the plant bends searching for the sunlight. All those setups will be done and kept. You have to identify it. So that is experiment number 15. Next 16 is enzymatic action of salivary amylase on starch. So salivary amylase will be added to starch. What happens to the starch when salivary amylase reacts on it? So all that you'll study under experiment 16. Next experiment 17 is to study the effect of temperature on the activity of salivary amylase. All these are biochemical related experiments. Next experiment 18, you have to test the pH on the action of salivary amylase. So acidic pH of the salivary amylase, basic pH, neutral, so different pH we have to study under experiment 18. So salivary amylase is nothing but produced in the salivary glands. So that is very much needed for the breakdown of carbohydrates. So whenever we are achieving food. So about that you will, some experiments related to salivary amylase. Next experiment 19, to detect the presence of urea in the urine sample. So how much amount of urea, whether it is there or not, we will be finding out. Next 20 is to test the presence of sugar in the given sample of urine. So all these are needed, like when you go for a checkup, all this will be done right in the by the pathologist, your urine sample, how much glucose is there, how much urea is there, all that they'll find out. So same thing you will be doing in the laboratory. So how much glucose is there in the urine sample is experiment 20. Next, how much of albumin is there in the urine sample? That is experiment 21. And how much of bile salts are there in the urine sample? It is experiment 22. Next, experiment 23 is you'll be studying about the human skeleton. So how total number of bones, how the bones are arranged, and what are the different types of bones, all that you will study here. Next, 24 is to study the joints. So once you know about the bones, you'll be studying about the joints in the human skeleton. So therefore, a total of 24 experiments you'll have to perform and study for first PUC biology practicals. So this is about the experiments. Now talking about the question paper. So here experiments is completely for A to F, that is it is for 20 marks. Viva is for 4 marks, that is 30 questions will be given in the forum book. From that you have to choose the Viva, and practical record is for 6 marks. The total it is for 30 marks. So this is about the first PUC practical examination. Next, talking about the question paper pattern. So 2 hours will be given wherein you'll have to perform the experiment and write wherever necessary and draw diagrams wherever necessary. Maximum marks is 30. So first is they have given like 2 experiments are given, which is for 5 marks. In that it is again a chit system. So when you pick a chit, which experiment you get from these 2, that you have to perform and write about it. So first is you have to take a transverse section of the given plant material A1, identify that different types of tissues in that or cells in that, and you have to write reasons regarding it. Next is prepare a temporary slide of epidermal peel, wherein you have to study the stomatal apparatus. So that these 2, out of these 2, any one you will get based on chit, whichever chit you pick. So it is for 5 marks. Next, question number 2, identify the given plant B1 and write the scientific name. So here knowing about binomial nomenclature is very, very important. You have to write the correct scientific name according to the rule, and you have to write any 8 characters of that particular plant. So this is about question number 1 and 2. So here also choices are there. 3 choices have been given. Identify B2, given plant B2 with its scientific name. Derive its floral formula. You have to write the floral formula as well as the floral diagram. All this you'll study in theory as well as practicals also. Next is identify and comment on the type of racemose or cymose inflorescence. I told you, right, there are different types of inflorescence present, meaning how the flowers are totally arranged in a plant. So here either it is racemose or cymose you have to identify, wherein 2 materials will be given, B3 and B4. 2 materials also you have to identify. So this one question will carry 5 marks. Again, this is also again chit system, wherein whichever experiment you get, that you have to perform. Next, question number 3 is biochemical test, which is common for everybody, wherein either urine sample, a carbohydrate sample or a protein sample, or fat, all that will be given to you. You have to put the appropriate chemical and you have to test whether it is present or absent based on the color or foam formation based on that. So that is about question number 3, which carries 3 marks. Identify the experimental setup D. Here, experimental setup will be prepared and kept. Only thing is you have to go there, see what it is, write what the experimental setup is, and you have to write the principle involved in that particular experimental setup. Say for example, the plant is bending towards right. Why is it? What is the principle behind it? So like that you have to write the principle. Next, 5th question is, so this is also for 3 marks. 5th question is identify the given slide of specimen E and comment. So slides of specimens will be kept, slides under the microscope and specimens in specimen jar bottles. You have to identify the specimen, write what it is, and then you have to comment on it. That will fetch you 2 marks. So identification is 1 mark and writing 2 points to 1 more mark. Next, again E also, identify the given slide of specimen F and you have to comment on it. There also it is for 2 marks. Next, Viva, wherein out of the 30 questions that are given in the biology forum book that you have to study, from those 30 questions, questions will be asked, 4 questions will be asked. If you answer them correctly, it will fetch you 4 marks. Next, practical record, if it is neatly binded, orderly maintained, experiments, everything completed and written well, it will give you 6 marks. Or if if you complete 75%, your marks will reduce. Again, 35% your marks will reduce. If you don't submit, you will not get any marks. Or maybe you will not be allowed to write the practical exam as well. So this is about the practical exam question paper. So I hope you understood this very well, wherein now you know what textbook you have to follow and what is the syllabus under it, and you know how the theory question paper pattern is, and you know about the practical experiments and also the practical question paper pattern. So with this, the introductory class comes to an end. So after this, we'll start with studying about each of the topics under chapters and the MCQs that can be asked from various topics we will look into. So see you in the next session, wherein we'll start with studying about the various chapters. Thank you.